Great. Well, thanks a lot for the invitation. It's really been a pleasure to come here and to listen to these great talks this morning. And so now I want to take the opportunity and tell you a little bit what we are up to in my lab. So we are interested in figuring out kind of the elementary process of chromatin organization and regulation. And really the key goal is to get from a this kind of genomic style view to a more molecular view of chromatin processes. And of course, chromatin is really involved in all regulation from, for example, differentiation of cells to kind of different subtypes, etc. So it's an elementary process and you need to know more. And so my lab is applying different methodologies to approach this, from chemical biology, where we can introduce post-translation modifications into histone proteins, to then reconstitution of systems to look at, interactions of factors with chromatin fibers, over structural studies to kind of in vivo, single particle tracking. And our questions really, involve, for example, how is chromatin structure regulated? And so you have, of course, a part of chromatin which exists in this open and active euchromatin state, and then other parts that are repressed, that appear as more compact heterochromatin, potentially restricting access of the transcription machinery. And a part of all these regulatory processes are, of course, post-translation modifications of the histones that involve from acetylation or methylation down to larger modifications like ubiquitination, where whole proteins are attached to side chains of histones. And then, of course, that interplays with a whole host of reader and writer proteins, as well as the transcription machinery. And so we are interested how all these things kind of interplay. And one core modification that I already mentioned that is kind of really close to my heart is ubiquitination. Just as a very quick recap, ubiquitination involves enzyme cascade. For example, so this E1 enzyme actually activates ubiquitin and then transfers ubiquitin as a thioester onto a ubiquitin conjugating enzyme, E2. And this E2 enzyme then collaborates together with an E3 ligase or a ubiquitin ligase to recognize a specific substrate, and then ubiquitin is transferred. And this process of ubiquitination plays a key role in chromatin regulation because histones are, have many different ubiquitination sites, and they have kind of very site-specific activities. So, for example, if you ubiquitinate histones down here at H2A, lysine 119, that leads to gene repression through the Polycomb system. If you put the ubiquitin up there onto H2A, lysine 13 or 15, it's a signal involved in DNA damage repair. Or if you move it over here to H2B, it's a signal for gene activation. So ubiquitination has very context-specific signals. And we are interested in all of these processes. But today I want to mostly focus onto this modification here at lysine 119, which is Polycomb gene repression. So in Polycomb, this is a very important regulatory process that is very important for development. For example, conserved from flies to men, there is a series of genes, these so-called Hox genes, that define the posterior anterior axis of the embryo. And Polycomb repression controls which of those Hox genes are activated in which segment of the body. And if this Polycomb repression system fails, then you get these segmental transformations where a gene that, for example, only should be expressed in one part of the embryo is now expressed throughout the whole embryo, and other effects like that. And that, of course, results in failure of development generally. And if you have mutations in the Polycomb system in an adult organism, that, for example, might lead to cancer. So what is the Polycomb system in detail? So this involves kind of three basic, enzyme complex. Of those exist many variants. So the Polycomb repressive complex 1 is an ubiquitin ligase that deposits this ubiquitination at H2A 119. This then provides a landing site for Polycomb repressive complex 2, or PRC2, which is a methyltransferase that methylates a very specific residue in histone H3. And this then, again, serves as a landing site for more PRC1, which finally kind of coats chromatin and potentially is involved in compacting segments to turn off gene expression. The ubiquitin signal, however, has also to be turned over by a specific deubiquitination enzyme called PRDUB. And so all these systems have to work together to get proper Polycomb repression. And so I want to just kind of show you, give you quick insights in all of these complexes. Firstly, starting with PRC2, this is a methyltransferase. And for example, Nogales' lab has shown that PRC2 can really bind to ubiquitin and therefore get recruited via accessory subunits. And in a collaboration with the lab of Jürgen Müller, we have also shown that PRC2 can contain specific DNA binding domains that recruited to certain regions of chromatin that are also often non-methylated. And so that kind of localizes PRC2 at certain regions and lead to further repression. Why do we have, and so this is kind of a primary reason why you need ubiquitination, right, to get these complexes there. But why do you need to turn them over? So you might imagine more ubiquitination might result in more PRC2 and then more compaction, more kind of silencing. But nature has evolved these deubiquitination enzymes that are important for ubiquitin turnover. So what is the reason for that? And so the lab of Jürgen Müller, our collaborator, and in particular postdoc Jacques Bonnet, they have observed that if you take wild-type flies and you look at the levels of H2A ubiquitination throughout the genome, you see some baseline level of ubiquitination. So then you can knock out PRC1, and so now you get rid of all the H2A ubiquitination in these embryos. And if they now stain for a particular gene, so this is abdominal B that only gets expressed in a certain part of the embryo, even if you get rid of a key part of Polycomb, you still get the proper localization of this gene. However, if you get rid of this deubiquitination enzyme, this PRDUB, then suddenly the gene gets de-repressed and it appears in other regions of the embryo where generally it should be silenced. So this is kind of inverse effect as you would imagine, because you would think more ubiquitination gets more repression, but here, more ubiquitination, we lose repression. And we also see that this is associated, so this is this abdominal B gene, with huge levels of H2A ubiquitination. So what is the mechanism for this? And so we work together and to solve this problem, and we have had kind of a hunch that, so this is a cryo-EM structure of a compact chromatin fiber. And you can see here, nucleosomes tend to stack in these kind of face-to-face stacking conformations inside these tetranucleosomal subunits. And if you now look at the actual structure here, here would be the attachment points for H2A ubiquitination. And you can imagine that if you now load this fiber up with lots and lots of ubiquitin, maybe you might actually inhibit the fiber conformation. And so that was the question that we wanted to test. And for this now we need kind of defined H2A ubiquitinated chromatin and then a method to observe what's going on. And so over the years, my lab has developed several methods to ubiquitinate proteins, and actually also the lab where I did my postdoc, Tom Muir's lab. And so we can now do it in many different ways. So this is one way where we ligate histones together out of several either synthetic or expressed proteins, ending up with a ubiquitinated histone. We can also use click chemistry-based protocols to get ubiquitinated histones. We have used that to make ubiquitinated H1. Or we can use, and this is a method that has been developed by Champak Chatterjee, a postdoc in Tom Muir's lab when I was there, and I have used it over the years, we can also use a disulfide-directed method to get ubiquitinated histones. And this has the added value that you can also chemically deubiquitinate your proteins if you want to. And so this is what we used. And so then to observe what's going on, we generally use FRET, for example. So we can introduce a donor and an acceptor dye into our chromatin fibers. And when chromatin is compact, we observe FRET. And when it kind of opens up, we lose FRET. So we can directly measure a structure, but it also gives us dynamic information. And actually, we could use that to look at how fast chromatin fibers can compact from an open state. And it takes, hundreds of milliseconds. And we could also look at the internal dynamics of chromatin fibers here in a collaboration with the lab of Klaus Seidel. And this allowed us to get a structural and dynamic view of chromatin dynamics. And it turns out these compact chromatin fibers are highly dynamic. They can exchange structural states in the sub-millisecond timescale. And then in the kind of a millisecond timescale, they can completely unfold and refold. And so this is a highly dynamic system. Okay, so what happens when you have now H2 ubiquitination? So first we look at chromatin in the absence of the modification. And what we can do is we can add salt, in this case 4 millimolar magnesium. Now chromatin fibers stack together and we see this high FRET peak or this high FRET readout. So that indicates that chromatin is compact. If we now have fibers that contain lots of ubiquitin, this high FRET peak here is completely gone. So H2A ubiquitination at these high levels prevents chromatin stacking. And if we use reducing agents to deubiquitinate our chromatin, we get the FRET peak back. So it's really completely dependent on the modification of H2A. So in vitro, H2A ubiquitination levels prevent chromatin compaction and thereby potentially also could prevent silencing. So if we go back into the cell, we can actually use a method called ATAC-seq to look at accessibility of chromatin. And we see actually in these embryos where we get rid of this PRDUB, the deubiquitination enzyme, we see higher levels of intragenic H2A ubiquitination here. And this results, here's ubiquitination, here is higher access. And we can correlate more H2A ubiquitination with more accessible chromatin regions, and the derepressed gene falls in this quadrant. So it basically tells us that if your balance of ubiquitination is out of whack, you lose repression and you poison the system basically. So you need a little bit of ubiquitination to start Polycomb repression, but if you have too much, you poison the system and you lose repression again. So it's all about the balance. Okay, now how do we get H2A ubiquitination there in the first place? And I mentioned this is the work of this enzyme complex, PRC1, and there exist many different versions of this, and in particular, the non-canonical PRC1 or variant PRC1 complexes are responsible for the initial deposition of H2A ubiquitination. And they have basically a domain called RYBP with which they can bind nucleosomes. They then have the ring 1A or ring 1B, mostly ring 1B subunit, which is part of the ubiquitin ligase domain. And then they have these accessory subunits of which there are many different versions. And, this PRC1 complexes, they can fold with any one of those different accessory subunits. In variant PRC1, it's mostly PCGF1 and 5. But 3 and 6 play also a role, and 2 and 4, they are shared between canonical and non-canonical PRC1. And so what we were wondering here is, how does PRC1 actually ubiquitinate chromatin? How can it recognize chromatin sites? How is ubiquitin transfer happening? And so again, right, PRC1 has to bind to the nucleosome, then it has to recruit an E2 that carries a ubiquitin, and then the ubiquitin is transferred. And so we wanted to observe this process in detail. And so again, we can reconstitute chromatin fibers, and then we put them into our single molecule microscope, and we can observe directly the interaction of PRC1 proteins, and then potentially the reaction that happens. So first, let's look at interaction binding of PRC1 complexes to chromatin. So here, each spot is a single chromatin fiber. Then we change color. Here, now we would see PRC1 complexes that are in dynamic exchange, and we can observe these dynamic interactions. And so high fluorescence means a PRC1 complex is bound, and then here it would fall off. Another complex comes in, it falls off. So we see this highly dynamic interaction. And that's of course important because PRC1 has to sample and scan the whole chromatin landscape to find its target sites. Now the question is, which one of these events, these long events or short events, which one of those are actually coupled to catalytic activity? So to actually observe that, we had to add a new color. And so now here we add a ubiquitin conjugating enzyme that's preloaded with ubiquitin that carries now a fluorescent dye in a different color, so in green. And we can now correlate the chromatin positions, the bound PRC1 molecules, and the ubiquitin transfer reactions in real time. And so now in these movies, we zoomed into individual chromatin positions. And so you can see PRC1 is in magenta and ubiquitin in green. And you will see, so there are some PRC1 complexes coming in and off, and then ubiquitin gets deposited and builds up over time. Now, to make sure that this is actually a catalytic reaction, we also repeated this with chromatin, where we knocked out the residues that get ubiquitinated. And here we now only see binding of PRC1 molecules, but no associated transfer because the landing site has been eliminated. So we can now take these traces and we can observe the reaction, the stepwise reaction. So again, in magenta, you see PRC1 binding events, and in green, these kind of stepwise successive ubiquitination of chromatin. So each step is another ubiquitin molecule that gets deposited. And sometimes we also see steps down, which we attribute to photobleaching of the dye on ubiquitination. And so we can generate from these traces, we can generate kind of chymographs over our whole field of view. So each line is a chromatin position, and you can always see some PRC1 complexes come and go, and at some point ubiquitin gets transferred and builds up over time. And again, if we get rid of the site that it actually gets ubiquitinated here, then we see much less ubiquitination. However, of course, histones have many lysines, and eventually one of them gets hit non-specifically. Importantly, if we only use E2 ubiquitin, we really basically see nothing. So you need the ligase for this. So now we can kind of tease apart the kinetics of the system. And so what we now look at is all these events of binding, PRC1 binding, that are associated with a step of ubiquitin transfer. So what can we say about this? First, how long does PRC1 need to bind to chromatin for a reaction to occur? And so if we take histograms of the lifetimes of all the active catalytic events, we see that they take about 20 seconds or so. So if you have very short events, usually that's not enough time to get the ubiquitin transfer. It needs to be bound in average about 20 seconds. How long does it take from the beginning of a binding event in average for a ubiquitin getting transferred? So we can also make histograms out of that, and we generally get about 10 seconds. If we increase the concentration of E2, then we can push this down. So here we double concentration, and so we go down with the delay, right? So this is a concentration-dependent mechanism. Then how many ubiquitins get transferred per step? So we can look at all our events and make histograms. So here you would see mostly one ubiquitin gets transferred per binding event, but with an almost same likelihood, you can also get 2 steps in one binding event. 3 steps then becomes already quite much lower likely, and all the other higher order reactions rarely occur. So it's a weakly processive process. 3 and more events are really becoming rare. And so we can interpret these 2 transfer events in the following way. So our PRC1 complex can ubiquitinate one side of a nucleosome, and then it can stay bound via its RYBP domain, reorient, and hit the neighboring nucleosome before it kind of falls off. So that would be an interpretation of how two events can happen. So now we can also look at the delay times if we have two steps or more. So we have one transfer, how long would it now take for the second transfer to occur? If we take histograms of this, we actually see kind of this, dual exponential behavior with a maximum time, and that generally indicates that there is a hidden step that we don't observe directly. And, this hidden step, we don't exactly know what it is, but it could be either the exchange of an E2 that is now discharged with a fresh E2 that carries a new ubiquitin, or it could be the time it takes for our PRC1 complex to reorient within the chromatin fiber. But either one of those, of course, accounts for this kind of delay. And so you need to wait for about 6 seconds before the system is kind of reset and you can go for our next ubiquitination transfer. Okay, so now we have a system where we can really look at the details of this reaction. And now one thing we wondered is, how does the PCGF unit, of which there are so many different variants, how does that affect the ubiquitination efficiency of PRC1? And so we compared PCGF1, which is one of the key subunits for the canonical PRC1, that's very active, versus PCGF4, which is associated with both canonical and non-canonical PRC1 and has been reported to be much less active. And actually we can reproduce that if we compare two complexes with either one PCGF1 or PCGF4, then you see the PCGF4 containing complexes give rise to much less ubiquitination. So what is the reason for this? So if we now take PCGF4 containing complexes, we can again perform the experiment. We see again individual ubiquitin transfer reactions. So it works quite well, but we observe if we compare this PRC 1.4 versus PRC 1.1 that we measured earlier, that it takes longer for the PCGF 1.4 to kind of deposit ubiquitination. So what is the reason? Now we can go back into the details of the experiment, and it turns out all the parameters are more or less the same, but the one thing that differs between the two complexes is the number of attempts PRC1 has to do before a ubiquitin transfer can occur. So the PCGF1 containing complexes kind of are very active. Even at the first attempt, your ubiquitin gets transferred. But in this case, you need to attempt many times. And we can quantify that. In average, for the PRC 1.1, it's about 2 events until the first transfer happens. For PRC 1.4, it's about 7 events. So this E3 ligase seems to be less efficient in hitting the chromatin right at the correct conformation to kind of allow ubiquitin transfer. And so this is something that we are currently investigating. So this is a model of the complex, in this case for PRC 1.1, and you see the complex here, and it has these kind of extended alpha helices that are not there for PRC 1.4, and we are investigating if this might be the reason, or if some contacts between the the E2 ubiquitin and the E3 ligase might be the reason for this reduced activity. So overall, we can now build a model of the whole system. So the PRC1 complexes, they search chromatin. They kind of end up in these very short-lived, non-specific states, or the kind of specifically bound state that lasts about 20 to 30 seconds. From there on, PRC 1.4 is much less effective in ubiquitinating chromatin. And this might actually be a good thing, because it is also part of this canonical complex that's important for compacting chromatin. And you don't want to have too much ubiquitination in compacted regions. But when it binds to the nucleosome, it has then a chance of affecting a first ubiquitin transfer that takes about 10 seconds. Then it reorients, that takes a couple of seconds to find a new nucleosome. It can deposit the second ubiquitin molecule, and then eventually it dissociates and can find another target. And so we hope that with this we can now observe, and really dissect chromatin readers and writers in more detail to get a molecular picture of chromatin regulatory processes. And with this, I want to thank the people who've done the work. I want to mostly acknowledge Alexandra, who was instrumental in putting up these single molecule experiments that are extremely complex with the PRC1 complexes. And our collaborators, in particular, a group of Jörg Müller, with whom we're collaborating for already a very long time on Polycomb. And of course, the rest of my group. So this is what we do in our spare time in Switzerland. And then, of course, our funders and you for your attention.
It's clock, and we have 5 minutes for a few.
I would have a technical question, Beat. So for some of your binding events, even the ones that were longer, so that shot noise should not be limiting, the brightnesses were sometimes quite different. Do you know what the origin of those differences in brightness is?
Yeah, so we have, of course, some ideas. I mean, first of all, it might be that, the chromatin templates are quite large, so we could be at different positions in the TIRF field. It could be that the dyes are in different environments, but we haven't been able to systematically figure that out yet.
Yeah, the chromatin is long enough, you could actually have...
Right, so of course, the fiber is maybe 200-300 nanometers.
Yeah, what are the prospects of adding even more colors and looking at the dynamics of the chromatin fiber while the reaction is happening?
Yeah, this is something we could do, and we have discussed it. And in principle, we have additional colors in our system. Yep. And so it's the question is how feasible it becomes, because already these experiments are quite challenging because we work with tens of nanomolar of components floating around in our TIRF field. But theoretically, it could be done by combining FRET and single molecule co-localization. Mm-hmm. Maybe if I find a brave enough collaborator to try that.
All right, if there are no more questions, let's thank Beat one more time.